Hello, my name is Thomas Rivet, and me and Dr. Berger have been working on site-directed mutagenesis and a reverse genetic system for Phi-6 on segment S. A little bit of background information on our virus. Our virus, Phi-6, infects a known plant pathogen known as Pseudomonas syringae. Phi-6 contains 14 proteins, 12 structural proteins, and 2 non-structural proteins. A non-structural protein is a protein that is encoded by the virus but is not part of the virus itself. The 2 non-structural proteins in Phi-6 are known as P12 and P14. Phi-6 also contains a 2-protein envelope that encloses a 3 double-stranded RNA genome. The 2 protein envelopes are known as the nucleocapsid and the procapsid, the nucleocapsid being the outermost protein envelope and the procapsid being the innermost envelope. The 3 double-stranded RNA genome is named based on their size, S being the smallest, M being in the middle, and L being the largest. Our objective for this research is to find out what exactly P12 does in Phi-6. Protein P12, as we know, has been studied to have a role in the virus assembly, but our goal is to see what part of the virus is assembled due to P12. Our first method was our primer design of P12. So we call these primers truncated. As we know, P12 is located on segment S of Phi-6, and substitution base changes were made 6 times throughout P12 randomly. We spaced them out from the beginning of the protein sequence towards the end. So on the table you can see there is truncated P12 numbers 1 through 5. 1 is going to be at the beginning of the protein sequence, and number 5 will be last in the protein sequence. And as you can see here, the lowercase letters in the table show where we did our base change. Now, these base changes were made from a start codon to a stop codon. So what we're going to see here is either no protein at all, or we're going to see incomplete proteins. Our next method was PCR amplification site-directed mutagenesis. This method was followed from a protocol given to us by New England Biolabs. And instead of a full reaction of 25 microliters, we decided to use a half reaction of 12.5 microliters in case we needed to do the reaction again. These primers were introduced to the plasmid, and through site-directed mutagenesis, they were able to attach to the site where they are encoded for the plasmid and do point mutations. The annealing temp for this was 65 degrees Celsius. After site-directed mutagenesis, the plasmids were then introduced to E. coli, where they were amplified many times to get many copies of the new plasmid. In this method, we used a reverse genetic system. Here we used truncated plasmids of S along with plasmids M and L were electroporated into competent cells. These competent cells contained a polymerase known as T7, which will be able to transcribe the DNA. This transcription will allow proteins to be made and the virus to be made within the competent cells creating the virus. Our next method was LM2691 competent cell preparation. LM2691 is our Pseudomonas syringae containing T7 polymerase that were grown and prepared for electroporation. These cells were grown at a steady state growth for 48 hours. After that, a culture was taken from the steady state to prepare for an exponential growth, and so that they can reach an optical density of 0.2 to 0.5. Once an optical density of 0.4 is reached, these cells were then centrifuged. After centrifugation, these cells were then resuspended in sterile water, and then cells were then centrifuged again. After this, the cells were resuspended in 10 to 25% glycerol and then centrifuged. This occurred twice. After that, a 1/100th dilution was made until an optical density was seen of 0.8 to 1.2. After this, cells were taken in 42 microliter aliquots, and then they were stored in liquid nitrogen for later. Our next method was electroporation of the competent cells. The purified plasmids and competent cells were placed into the same test tube. Each test tube contains a different truncated P12 plasmid 1 through 5. These test tubes were then left to sit on ice for 1 minute. Mixtures were then placed into the electroporator set at 2,500 volts. After electroporation, cells were then set into the shaker at 37 degrees Celsius for 1 hour for recovery. Now, what happens in the electroporation cell process is once the cells are electroporated, they are created holes in the membrane, allowing these plasmids to slip through into the cell. When they are left on recovery, these cell membranes fuse back together, enclosing the new plasmids into the cell and allowing for transcription to occur within the cell. After electroporation, we did a bacteriophage plaque assay and plaque isolation. So 100 microliters of these recovered competent cells were mixed with top agar and poured onto a media plate containing tetracycline. Now, the reason we do tetracycline is because that with these plasmids, they are tetracycline resistant, so they are able to grow on tetracycline and nothing else is. These media plates were then left for 48 to 72 hours to obtain plaques. If there were any plaques, they were isolated and analyzed. They were isolated by groups, so that we wanted to take the most isolated ones away from grouped plaques to pick with a 1 milliliter pipette. These were then placed into LB broth around 100 microliters, and then they were set in the fridge. What we're looking for here is any differences in size or shape of the plaques. Due to unseen circumstances of COVID-19, we were not able to finish our research. But the conclusions that we do have is for the bacteriophage plaque assay, HB10Y PD was able to grow plaques of unmutated virus, whereas HB10Y ATCC was not able to grow. The only difference of HB10Y PD versus HB10Y ATCC is where we got the cells from. LM2691, as you recall, is the competent cell that contains T7 polymerase. The problem that we had with this cell is that it did not grow on our tetracycline plates, which was a big problem. We needed these cells to be able to do the electroporation. We also had plasmids of truncated P12 numbers 2 through 5. They were able to be recovered after purification, whereas number 1 was not able to. Where we stopped was after plasmid purification, where we got the truncated P12 numbers 2 through 5, and we're not able to get truncated primer of P12 number 1. So what needs to happen now is we need that growth of LM269, the HB10Y PD with T7 polymerase. That needs to occur for electroporation to occur. So after that is grown, there needs to be the electroporation of these plasmids into the competent cells, which then will result into the bacteriophage plaque assay and isolation. And after that, what we need to do is do a sequencing of these, of probably just segment S, to see if after recovery and after plaque isolation that sequence is still there that we made. We would like to thank Trine University for giving us funding for this research and also allowing us to use their labs on campus. We'd also like to thank the New England Biolabs for giving us protocols and supplements to be able to do this research. And we would also like to thank the Mindage and Danthe Labs for giving us supplements and provided equipment for us to finish this research.